Team Antidote, analyzing new treatments. Once again. Good evening, we are Team Antidote, analyzing new treatments for infectious diseases through obtained therapeutic extracts. My name is Rahel, and I'll be presenting on behalf of my team, along with Nathan and Angela, about our research and the progress we've made thus far. So we're going to begin our presentation with a background on HIV and HCV, as well as our plant, Philanthus neurari. We will then talk about our research questions and our justification for doing this project. We will touch on our hypothesis, our team composition, as well as our methodology and progress with regards to genetics, biochemistry, and cell assays, our goals and future directions, and then a quick wrap-up. So human immunodeficiency virus, or HIV, and hepatitis C, HCV, affect the lives of over 200 million people around the world, highlighting the epidemic status of these diseases. To make matters worse, 30% of those people who are infected with HIV are also co-infected with HCV. This condition is called HIV-HCV co-infection. Today, scientists are under enormous pressure to develop treatments for HIV and HCV that are both effective and tolerable. There are currently very few treatments that are available for HIV-HCV co-infection. Cocktails of pharmaceutical drugs are often used, but they only target HCV monoinfection. The only FDA-approved treatment for co-infection at this time is the use of pegylated interferon alpha-2A plus ribavirin. This treatment specifically targets cell receptors of infected cells and inhibits viral replication. However, this treatment is entailed with a number of negative side effects, including insomnia, cognitive changes, depression, irritability, skin rash, hair loss, weight loss, as well as anorexia. And now Nathan will talk about how we decided to take on our project.
Thank you. Okay, so our team decided to approach the HIV-HCV issue from a different angle. We asked, what if we researched the potential of herbal medicine? From this perspective, we came across our plant, Philanthus neurari, which you can see is on the table. This plant is native to tropical areas in South and North America and India. In these areas, it is used as traditional folk medicine, where it is boiled into teas and then used to treat ailments such as jaundice, kidney stones, and loss of appetite. However, recent scientific studies have focused mainly on the liver-protecting activity and the antiviral activity of its extracts. So various studies have shown that there is indeed a correlation between the use of Philanthus neurari and the restoration of liver health. In India, there was a clinical study done where liver function returned to normal within 5 days in children infected with acute hepatitis after they were treated with the plant. There were also an in vivo study which mice pre-treated with the plant were induced with liver cancer and had double the life expectancy of those mice that weren't given the pre-treatment. So the two main chemicals that are active and known to help the liver-protecting agents are the phytochemicals phylanthin and hypophylanthin. Philanthus neurari extracts have also displayed antiviral activity against HIV replication. When an alkaloidal extract from the plant was tested on HIV-infected cells, the overall result indicated that viability of the cells was extended and it reduced the rate of cell death from HIV. Subsequently, a different study discusses the properties of a specific compound, neuroside, which comes from an aqueous extract from the same plant, that inhibits the binding of certain HIV proteins, which prevents the virus from replicating. Aside from neuroside, there are other various chemicals from Philanthus neurari that have shown the same effect of antiretroviral, which are proteins, terpenoids, xanthos, and polysaccharides. So another sister plant to our plant is Philanthus amorous, and it has shown the potential to block all 3 critical enzymes of HIV replication, which we know from bioclass are integrase, reverse transcriptase, and protease. And therefore, it indicates the capability of this family of plants being used as anti-HIV drugs. So after reviewing all this information, we came up with our 2 research questions. The first is what active compounds are present in Philanthus neurari, and how do these compounds affect the viral load and cell viability? Now for this question, it would be ideal to isolate and identify a specific active compound, because that's what's most easily turned into a pharmaceutical drug in our economy. However, this is an extremely complex process, so for our goals, it is more ideal to just locate a beneficial extract from the plant. Our second question is, what is the effect of chemical compounds found in the plant when combined and compared with the medicine used in conventional treatment, specifically T20 for HIV and interferon alpha for HCV. Although conventional treatment is effective to a certain degree, we want to find an alternative treatment that can either improve the current treatment efficacy or at least give those same effects with less severe side effects, but hopefully both. So our hypothesis is that the extracts from Philanthus neurari will have an antiviral effect on cell cultures for HIV and hepatitis C, as we will see through decreased viral load without significant loss to cell viability. The reason our research is novel is because this is the first time that this specific plant has been tested on an infectious cell culture model for HCV. In past models, the virus has only been replicated once, and scientists could not determine how to propagate a viable model for HCV. And also, it is important to note that the efficacy in co-infection, the treatment of co-infection of both diseases, can only be done in an animal model. However, we're hoping that by testing both diseases using the same method with the same plant, we can show that it's important to do more research with the use of this plant on both diseases. And now Angela's going to talk about our group dynamics and
All right, so to answer these research questions, we decided to organize our team into 3 major subgroups. The first subgroup is the genetic barcoding subgroup, and they're trying to document the specific species of Phyllanthus we're going to use. The chemical extraction subgroup will then take the plant and try and extract the active compounds. And finally, the cell culture study subgroup will take these extractions and test them on cells for efficacy and cell viability. Our team proposed a methodology that is comprised of 4 main procedures. The first is plant cultivation, followed by DNA barcoding, which will continue throughout the entirety of our research. The other 3 procedures are chemical extractions, cell culture studies, and chemical analysis, which will be dependent on each other in a linear fashion. So before our research could begin, we needed plant matter. So we obtained seeds from an online vendor, and then we germinated the seeds at the U.S. Botanic Gardens growing facilities. They were then transported to the University of Maryland greenhouse for further cultivation. After cultivation, we harvested our plants and then dried them so they could be used in barcoding and extractions. Since Phyllanthus neruri is taxonomically confused with other species within the Phyllanthus genus, there's a possibility that the samples we obtain won't be specifically of the neruri species. So in order to minimize these problems, we will use DNA barcoding to keep a genetic journal of the plants we use throughout our research. This might not necessarily identify our plants as Phyllanthus neruri, but it'll show that we are using the same species throughout our research. So DNA barcoding involves classifying new species based on sequences of certain genes in the plant genome. Segments of these genes are common across all plant species, while other segments differ enough to allow differentiation between species. So one of these segments is RBCL, and RBCL provides easily retrievable sequences across land plants due to its low variability, while MAPK, which is another sequence, provides a high level of discrimination between species. Combined, these 2 genes showed 72% discrimination of plant test cases. Additionally, the Consortium for the Barcode of Life recently reported in the scientific journal Nature that it had chosen RBCL and MAPK as the approved DNA barcoding regions for land plants. So currently, we are still optimizing our barcoding experiments. So far, we've conducted a few experiments to amplify the barcoding regions through PCR, but we haven't been successful yet. The problem with PCR is that it's often a tedious process, and there are various parameters that will affect whether or not your experiment is successful. So one of the biggest issues we've had so far in our research is finding the correct primers to do this experiment. So this gel up here shows one of our experiments to try and amplify the gene. The left shows the bands of ligers, which provide marking points to act as a reference for the size of the DNA. Ideally, if this was a successful experiment, you would see the same kind of bands in the other leg as well. And now Rahel will talk about chemical extractions.
Okay, so chemical extractions is the core of our research. So the first step is, chemical extraction, excuse me. We did use multiple solvents such as water and ethanol at varying temperatures to extract and target different chemicals within our plant. And this gave us a base of extractions to use within our study. We began biochemical extraction procedure with a methanol extract. 5 grams of dried plant matter were amassed out from 10 different plant samples of the same species. The plant matter was ground and suspended in pure methanol. This was then refluxed in a hot water bath for about half an hour, after which the liquid was filtered and collected separately, while the plant matter was refluxed again with 80% and then 70% methanol. The final solution was evaporated to obtain our first crude extract. This crude extract was then dissolved in 90% methanol. The new solution was then partitioned in a separatory funnel against hexane, chloroform, and carbon tetrachloride. Each layer was then collected and evaporated separately to yield a total of 5 extracts of varying composition. Now Angela will finish up with cell culture studies.
So once we've obtained all of our extracts, we will begin our cell studies to measure the efficacy of the extracts through cell viability, viral load, and possible synergy. And this will take place over the next several months. So for cell studies, we'll take the chemical extracts and test them on different cell cultures. So each cell culture will be respectively infected with their virus, either HIV or HCV, and then each experiment will include a negative control, which will receive no treatment, and a positive control, which will receive conventional drug treatment. Then each extract will be applied to a cell culture at a standard concentration until all the extracts have been tested. Then we will evaluate the effectiveness of the extracts based on 3 criteria. So the first is the viability of the cell cultures, whether they are dead or alive, and we will test this by the visibility of a specific dye we're going to use. Then we will test the viral load of each cell culture and whether the extract has reduced it, increased it, or kept it the same compared to the negative control. And this will be tested by extracting the viral RNA and then quantifying it using a fluorescence probe. Finally, our third criteria is the synergistic effects of the extracts. We're going to take each extract that we found effective in the previous 2 criteria and either combine them with each other or combine them with the conventional drug treatment. And we're going to see if this reduces the viral load or increases cell viability. So currently, our team has finished doing the majority of our literature review, and we've also started our DNA barcoding and extraction lab work. Over the summer, we completed 1 harvest of our plant, and then another harvest was done in mid-September of this semester. During both harvests, we cut the specimens, we labeled, masked, and then dried them. We were able to collect about 60 specimens. Our team also applied and received both HHMI and ACC-IAC grants. And with the help of these grants, we were able to obtain lab space at the University of Maryland Greenhouse. Now Nathan is going to talk about our future goals and timelines.
So our goals for the remainder of the project are continue with our lab work, complete data analysis, and begin our senior thesis. In summary, we basically just want to get our DNA barcoding to work and find the specific primers for our plant. Then we want to continue with our chemical extraction process so we can come up with as many different extracts as possible, and finally complete our cell studies so we can analyze the effectiveness of each extract. So then we have some advice for the freshmen. Okay, so just get organized early. That's the most important because no matter what project you choose or how organized you are in the beginning, you're going to run into obstacles. So the sooner you start, the sooner you'll be able to overcome these obstacles and move on. And you also really want to get in contacts. As you can see from each team, they all have contacts with experts or friends that really helped out with the project. And you'd be surprised how willing people are to help you out within the same field. And then just apply for grants and be willing to dedicate a lot of your time to the project. Okay, and so we have some acknowledgements. We'd like to thank our mentor, Dr. Andrea Otteson, HHMI and ACC-IAC for the 2 grants we received, and then the Gemstone program and the U.S. Botanical Gardens. Thank you.